My name is Helene Puccio, and I'm a research director working on recessive ataxias in Lyon, France. Part of the most exciting developments in the ataxia field right now is the fact, well, the first part is the fact that the first drug has been approved for Friedreich ataxia. So this is very exciting. So this is showing that we can make this way from preclinical all the way to clinic and into approval. And then the other excitement is that there are many different models, mouse models and cell models being developed that are showing common pathways among different ataxias that we could probably target. In ataxias, or in general in clinic, we talk about repurposing drugs. Is a drug that has a authorization for another disease, but that we can reuse it for a new indication. And so this is a way to accelerate research. So, for example, in the ataxia field, for example, ceftriaxone, which is an antibiotic, has been shown by Francesca Malteca to be actually promising drug approach for different cerebellar ataxias. And so repurposing this drug would accelerate the drug development if it would work. So it's right now in preclinical trials in mouse models. Many of us are doing screenings of FDA-approved libraries to try to find new drugs that are already approved to be able to repurpose them. So this is something common. And actually a lot of the antioxidants in the early age for FA were repurposed drugs. So working on ultra-rare ataxias is important because all ataxias are important, and there might be some common pathways. What we're doing in my lab on ultra-rare ataxias is we're generating mouse models and cell models, and then we're taking a drug repurposing approach to try to see if we can treat them. Because, of course, in these ultra-rare ataxias, pharmacological companies are not going to be developing new treatments. The first lab showing the proof of concept principles that gene therapy could be worked for Friedreich ataxia, both on the cardiac aspect and the neurological aspect. So this was extremely important and actually has enabled to get so many pharmacological companies interested in getting gene therapy for Friedreich ataxia. I'm continuing the work in the lab to try to find a way to have lower levels of frataxin to make sure we don't have toxicity. And so I'm continuing to develop this, and hopefully this will help the development by pharmacological companies to put it into clinical trials. During my PhD, I worked on muscular dystrophy, and I worked on gene cloning, so finding new genes and finding mutations for these diseases. And after that, I wanted to go into the post-gene area, so try to understand, once we have a gene, can we develop models and find therapeutic approaches? And it happened to be that the Friedreich ataxia gene had just been identified, and so there was a need to develop models for this disease, and that's how I got into it, and I've been there since, and working on ataxias and making it part of my family. There is many different labs, many more labs than used to be working on ataxias and being interested in ataxias. There are many clinical sites that have been developed and that are becoming better adapt to clinical care and helping patients. And there's an enormous amount of pharma companies being involved in trying to develop therapies. And so I think that it's a good time now. It's much better now than it used to be because there's more chance of getting a therapeutic approach, and there's also more chance of getting better clinical care. I think the patients should be optimistic that things are going, but we know that the research that we're doing is always too slow for the patients.